Okay, well, welcome everybody. Thank you for coming to this talk. It's quite interesting. I've had to modify it slightly, but basically, as you'll see from the title, and thanks Emma for the introduction, I'm one of two grassland specialists within Natural England. But the reason I'm talking about wax caps is because a couple of years ago I said I would like to do the grassland fungi bit as an addition to my role. So I'm really a grassland ecologist or ecologist, and an imposter in terms of my mycological expertise, but I just happen to really enjoy going out surveying wax caps and their associated fungi. So that's where I'm coming from. And really today, this is kind of a look at where we are in the sort of early 2020s with the conservation status particularly and the distribution of grassland fungi. So it's more a kind of conservation talk as opposed to a mycological one, but obviously there'll be linkages. So without further ado, I'm going to go to the next slide, hopefully. Already it's not working. Okay, great. So I'm going to talk about what waxcap grasslands are, and I'm sure a lot of you on the call will know exactly what waxcap grasslands are. But very briefly, what we're looking at here are the fungi that are predominantly associated with grassland community types in the UK. And also, dare I say, most of Northern Europe as well. What we're not talking about are the fungi such as the parasols, the dung fungi, horse mushrooms, etc. Those fairly generalist species. And of course, in America, the wax caps, for example, as other fungi, occur in a woodland setting. But we're really going to focus today on just the sort of the grassland fungi that occur, or the wax caps and other grassland fungi that do occur just in the grassland habitat. Okay, predominantly. I can't see exactly all that. Anyway, so for those that don't know, or just as a refresh really, what we're looking at is what are now termed the CHEG-D series of fungi. And in my collection box there is a nice selection of what I found on a survey last November. And they're large. The CHEGD stand for Clavariaceae, the clavarioids, the Hygrocybe, that is the wax caps, the Entoloma group, the earth tongues, the geoglossums, microglossums, trichoglossums, that broadly covers them. And then this slightly different group of, which is the D, which includes a few of the taxon groups, the Dermalomas, Porphylomas, although there's been some changes with that, and the Chamarophilopsis. There is further taxonomic changes in there, but I won't go into the detail of that. And in the simplest terms, the sort of more CHEG-D fungi, the higher the score you will have, the more important the site is. So there's no weighting to the score. One Clavaria, one Hygrocybe, they just equal 1 on a score sheet. Okay, so there's no weighting to that at all. So that's a kind of introduction to the CHEG-D system, if you like. And just to show everybody some of the species, a really lovely collection of what are, in English, clubs, corals and spindles. That name seems to stick now, and it's just a nice example. As you can see, they're varied. Some spectacular colours, such as the violet coral. Some species you'll find, if you're lucky, in your lawns, such as the white spindles and the meadow coral down here. And others are a lot rarer, such as the skinny club. And it's not actually Romariopsis kunzii anymore, I think it's Robusta. But like I say, I'm not going to apologise for the taxonomy tonight, because it's always changing. So you'll see the names that I last come across within the presentation. And then we have the Hygrocybe or Hygrocybe, which is kind of where the waxcap grassland term comes from, and a really colourful collection of species. We've got crimson ones, orange ones, splendid waxcaps here, and then also you've got the parrot waxcaps, heath waxcaps, the Gliophorus group, if you like, and they're a whole mixture of, as I say, oranges, yellows, purples, browns, fabulous set of mushrooms, sometimes termed the orchids of the fungi world. Then we have the Entomomas, the E, obviously, and actually this comprises the largest group of species found within the kind of waxcap grassland assemblage. They're a very tricky group, to put it mildly. I've shown here some of the easier ones to identify, such as the big blue pink gill, Entomoma bloxamii, which is actually a protected Section 41 species in England. I think it is for Scotland as well, and Wales. And things like the mouse pea pink gill, which if you leave it on a radiator will smell pretty horrible. But they are generally quite a tricky group to identify, and almost certainly take microscope work to identify them properly. Then we have the earth tongues, equally quite a challenging group as well, quite hard to find generally in surveys. They tend to pick out these mossy, shorter swards, and you have to sort of down on your hands and knees to find those, and again require microscopy to, by and large, identify them accurately. And then finally, this group of the Ds, if you like, with the dermalomas, these fan faults, and the meadow caps as well. And apart from Dermaloma cuneiformium, these tend to be quite rare, and if you do find them, you're probably looking at grassland fungi sites that are generally quite rich. I mean, these are obviously generalizations, but that's often the case. And interestingly, things like the mealy meadow cap in England, at least, seem to have a western and northern distribution, with I think the southernmost record of that species being in Somerset, but somebody may be able to tell me otherwise. So that's a kind of very brief introduction to the sort of Cheg D, the grassland, the waxcap and their allies group. So, go back. So in my role for Natural England with grasslands, we've been undertaking some work in the last few years, but particularly the last 2 years, reviewing the grassland waxcap site series. And there's several drivers for this work, and I've outlined them here. I'm sure you've heard the figure of the decline of grasslands, not just in the UK, but across Europe as well. And often the figure trotted out is 97% loss of flower-rich grasslands. Well, almost certainly whatever's happened to those flower-rich meadows, etc., will have also had an impact on the grassland fungi sites as well. Although the picture is slightly more complex than that, which I'll come on to. We're also very concerned about habitat decline, particularly actually decline in sort of traditional management practices. For example, grazing, livestock, and hay meadow management, that sort of thing. So that's the change in farming practices, if you like. A big thing that's come flying in very fast is the whole tree planting agenda, with the government having objectives and targets. So in the UK at the moment, each year there's a target to plant 30,000 hectares across the UK. And the big concern from Natural England's point of view and our other agencies is, where will these trees be planted? And dare I say, you can probably guess that there will be a lot of grassland sites that will come under pressure for delivering tree planting targets. Rewilding is another interesting one that's again come flying up the agenda. Now, whether rewilding will have any impact on grassland fungi sites is a bit unknown. If they keep grazing animals on a site, well, then hopefully they'll be able to deliver the sort of management that grassland fungi require. But again, this is all coming up on the outside lane, if you like, and we really don't know what the impacts. You can probably guess what the impacts might be, but we don't know for sure. Another key element is there's very, very few sites that are actually protected. I'll come on to that in a moment. And really, in terms of the last time a detailed assessment was done, we are looking, actually, the last report is getting on for 20 years old now, in fact. But a lot of the information that went into the report is a lot older than that. And finally, well, rather usefully, from a plant point of view, the plant people were effectively looking at a vascular plant database, with BSBI, and Natural England working with the British Botanical Society of Britain and Ireland to develop a heat map of where all the records for plants are. And that was a bit of a catalyst where we thought if they can do it for plants, we can do it for fungi. So we piggybacked a little bit on some of their work and then developed it for fungi ourselves. So when I started this project, in fact, I started it back in 2015 in real terms, but I did a full review of all the information that was out there. So we were looking at plant life's important fungus areas back in 2000, coming back to the report for the England review, which was done by Shelley Evans. That was the first kind of proper review, if you like, to assess the sort of status, the conservation status of waxcap grasslands in England. Then in 2012, an internal report was commissioned with Natural England Kew, actually, with Justin Smith, who some of you may have known, who sadly died a few years ago. But a report was done, but was never published, unfortunately, to look at whether or what sites might qualify for SSSI, for the Sites of Special Scientific Interest notification. That didn't really go anywhere. So in 2015, Natural England did an internal review to assess the SSSI series, the protected site series. And on the back of that work, grassland fungi came right to the top of the agenda when we realized actually they were incredibly, or shall I say, their representation within the SSSI, the protected site series, was extremely poor. I think at that point in 2015, we had certainly two sites, I think a third was about to be notified, that was actually protected for its grassland fungi. And I was fortunate enough in 2017-18 to be involved with writing the brand new guidelines, revised guidelines for fungi. And in fact, this was a complete revision of all the previous work. So there have been some guidelines in 2019 to inform how we would go forward with notifying sites. In 2018, the new guidelines were published, and I was fortunate enough to be asked to do the Grassland fungi chapter, which was a complete revision, and critically, a new kind of assessment was done based on some data that was coming in at that stage, to review the sort of CHEG scoring criteria. So that changed, and in fact, we made it harder, just slightly harder for sites to actually qualify to become SSSIs. So that now is the standard piece of work that we use for guiding us to notify SSSIs for the UK, not just England. So I'll give you a sort of update of what is happening now. So on the back of the 2015 review, I also then compiled a separate England grassland fungi database using the available data that we had from the Shelley Evans report, Justin Smith's work that I pointed out, but also any other open sources of data that were available. Interestingly, we used things like all the BMS field mycology journals and pretty much any other source of open information that we could find. And on the back of that, I compiled a separate England Gazetteer. We realized that wasn't enough. And so in 2021, well, actually in 2020, I commissioned, managed to secure some funding to commission a survey, an analysis of the BMS fungal records database of Britain and Ireland data. And as you'll see, that to compare that with the 2003-04 work that Shelley Evans did, with 20,000 records. In 2021, we were looking at over 60,000 records of the CHEG-D series. Sorry. So 3 times more data to help inform us for this database. So it was a major piece of work. David Mitchell, who some of you will know, was our contractor, and he did an incredible piece of work actually for very little money. So I will congratulate David while we're here now. I don't know if he's on the call, yeah, it's fantastic. So what we ended up with was this new database of grassland fungi. But we also realised that it wasn't going to be a complete assessment. So Claire's going to talk about the work that Sussex did, but we gave a contract to Sussex as well. Where I am in Dorset, we also did a smaller contract, and because I'd also done a lot of work with people within Dorset, Brian Edwards, who you may know I've done quite a lot of survey with, I've been submitting records to Dorset. So we looked at all the non-FRDBI there for Dorset. I've also been working with the Peak District National Park on various projects, and they've been feeding in data, which has been great. And the Northumberland National Park, but particularly, dare I say, one of my colleagues, Andy McClay, who's within our Natural England Field Unit, has provided actually the bulk of the records for North East England. And his work as a contractor for Northern water utility companies and then for Natural England has provided a huge amount of data for us. We've used a little bit of plant life information from our Waxcat watch. There was a lot of duplication with the records that were coming in, perhaps not unsurprisingly. But actually, we've managed to identify a few more sites from the data they've provided. And dare I say, social media is increasingly important to contact people that don't necessarily always submit their records to the various recording platforms. So I've been in direct contact with a few people on social media, which has been really, really useful. There's obviously some cleaning up of the data that's needed with that. So it's not perfect records, but it is leading us to sites that we would otherwise not necessarily find. And this is just a screenshot to show you one of the output from the database. So this isn't interactive, and I'm not going to show you how it works, but it kind of gives you the idea of the platform that was developed. And it's really showing how we've done our ranking, how we've done our assessment to kind of inform where the most important sites are in the country. Now, behind this database is all the data, obviously the 60,000 records that have been pulled into sites. Various other records sit behind this. But the good thing about this database, it's being able to transfer into a GIS mapping tool, which I'm hoping I'll have time to show you as well. But there's a huge amount of information behind this database. Like I say, unfortunately, I haven't got time to kind of go through this. Access isn't the best thing to illustrate, unfortunately, a lot of the time. Anyway, we'll move on. So what's in the database? We went through the 60,000 plus records, and there was a complex cleaning exercise. So just in summary, sites were merged, sites that were in the sea, for example, were removed or fixed. Confidential sites were removed. They were assigned for vice county and county. Where sites were known, they were matched with grid references. I'll explain that a little bit more. We weren't able, most of the time, in fact, to assign sites directly to fields or polygons, what we call polygons on a map. So there was a lot of sort of messing around with grid references, trying to see if we could assign them to the closest known point of where we thought the sites were. But a small number of impossible grid references were also removed. So there was a lot of cleaning that went on with this data. We ended up with what we've called preferred site names. Initially, the first analysis developed over 8,800 sites. When we did some further analysis of the data, we ended up with these 5,800 plus sites. Now, what that means is that we've now got 5,800 sites in England that have at least one Waxcat or their allies assigned to a site. Now, I'm not going to be looking at those records at one site. I will be looking at sites with 10 or more CHEG-D because there's just too much analysis to be done. There'll be a parking of that data, which we will then come back to at another stage to see if it's worth looking at those sites again. But we're doing a first kind of a tranche to look at those sites which we think are important, and I'll explain a little bit about that. We've assigned them to sub-sites and meta-sites. And as an example, the meta site, as a good example here, is the Peak District National Park. We've called that a meta site, and within the Peak District National Park, we've then called sites based on their exact details. So Allport Valley, a National Trust site that's been surveyed extensively by Neil Barden. It is the site that we're assigned as an important site. Okay, hopefully that gives you a rough idea of how we've done this. I'm having to keep moving stuff around, unfortunately, which is slowing me up. Now, the results that we've got, the data set scope, we feel from the FRDBI, from the records that we've got, in fact, we think now, because I've had some more records, that we've got about 70% coverage of the important sites from the FRDBI for England. From the data that I've compiled for our separate England grassland fungi database, the Natural England one, there is some significant differentials. I've laid it out there, which gives you a rough idea. And I realized when I was doing the slide, it's possibly not as clear as it should be. But what we're ultimately doing now is bringing all the data sets together to form one platform. And at the moment, we haven't quite decided whether that will be an Access database or an Excel spreadsheet. What critically it's got to do is got to be readable for our mapping exercise. With all this data coming together, we now think we've probably got 80% or more representation of what we think are the important sites in the country. Okay, I'll leave that figure there hanging for the time being. And I'll very briefly whiz through this because I know time is running away with me. The database has got all the sites with the CHEG-D scores and the triple SIs with CHEG-D scores. We've also used the IUCN species, so the red list species for IUCN. It catalogues the number of visits under the FRDBI and all the dates of the visits. We've got triple SI thresholds, CHEG-D thresholds, species tables, records tables. Importantly, for further users in the future, we've mapped them down to 1 kilometre squares and 10 kilometre squares. What we're hoping to do is then hone it down even further, where we can get more detailed information to the sort of level of field information or polygon information. But there are issues with that, not least of all to do with licensing and ownership, the land ownership details as well. So there's some stuff to be ironed out there. And another piece of work that was really useful because of all the FRDBI data, we've got a phenology of all the CHEG-D species as well. And just for the record, if you want to go out and survey, and I'm sure most of you realise this, October and November, if you can't do any other months, just do October and November. That probably goes without saying, I'm sure most people know that. We've also developed indicator species as well. That comes back to the high diversity, the HDI indicators. Again, I haven't got time to explain that, but in the triple SI guidelines, the JNCC guidelines, that is also described, the high diversity indicators in there as well. And just to finish, we are ranking as we go along the sites based on the CHEG-D. So that's an ongoing exercise at the moment. Excuse me. Now this map is a little bit meaningless, but I think in some ways it does show the widespread nature of grassland fungi sites in the country. This is the 5,000 preferred sites, if you like. Interestingly, it does show up gaps, perhaps a bit surprisingly. Those with geography, know the geography of England, might think, oh, Dartmoor, Exmoor. I'm sure there's many more on there. East Anglia, perhaps not unsurprisingly less there, but maybe again, people will be able to fill in gaps. And there are obviously gaps as well. But this is just the FRDBI, I put FRDI there, the FRDBI data, not all the other combined data sets. Okay, so it's important to just bear that in mind. When we run the analysis in terms of those sites that would meet the triple SI thresholds, again, this is changing. We have 152 sites that will pass the triple SI thresholds. So that means all these sites could potentially be protected one day. Now, that is an ambition of mine. If we ever get there, that will be a fabulous achievement. So I'll describe it a little bit. But what we do know that very, very few sites pass all the thresholds of the Chegg-D thresholds. Okay, Hadrian's Wall is a complex. Rowcliffe Manor is a single site. So Hadrian's Wall is one of the meta sites, if you like. This is very much a work in progress. We're developing what we call heat maps. And as more and more data come in, and just to kind of put it into perspective, we've probably, at this stage, we've only analysed about 30% of the records in real detail. So this map will be populated more. I realise I'm running over. And just to give you an idea of the sort of bias of recording, you can see that around 60% of the records that we have are the wax caps. So obviously everything else is lesser recorded. Hence, I suppose that's why we're calling wax cap grasslands. In terms of what's coming, a key element of this work is to look at whether we can notify more sites. Okay, that's a critical element of this piece of work from a natural England perspective. And earlier this year, I put forward 21 sites as a high priority for a SSI notification pipeline. And although I haven't got my 21 sites, we have got some new sites going in that will be notified within the next year or 2 years. So that's really encouraging. But also importantly, on those protected sites, we've also been able to identify from the database SSIs that have grassland fungi as an interest feature, but weren't notified that. As an example, Dolby Warren in Somerset, North Somerset, we found out that it's an incredibly important site from assessing the data. And the next stage will be to include those sites, to include them in the conservation or the condition monitoring for those sites. So that's an ongoing work. I am afraid I'm going to have to whiz through a little bit, but this kind of gives you a bit of an idea as still how rare internationally important sites are. You can see from the data, and how many more sites there are, obviously at the lower end of the height. This is just Herbaceous, by the way, okay? But we've set it out, and a lot more data is being added to this, so this will change. In fact, it's up to 900 now, I believe. We're developing the grassland database as we go on. Hopefully next year, one of the key outputs will be that there will be a public-facing new layer for the grassland fungi. And that the ambition is to get that onto the government's mapping page called Magic. And we're hoping that it will also become, in effect, a priority habitat. That's something I'm working with to present to DEFRA, the government's environment, if you like, the Farming and Environment Agency. And also we're going to develop an inventory with Chegg-D with 10 species or more to become a constraints map for the tree plot, for things like tree planting. Now that's again a work in progress. We're aligning all these sites with our priority habitat inventories, and we're also hoping to work a little bit more on the citizen science stuff with plant life. I don't know whether we're going to get time for this, because I know if we do, I'll come back to this, because it'd be really good to show people an interactive map, because there's a lot of work going on, and we're, dare I say, quite proud of this in terms of an interactive tool. And this is the sort of thing that you'll hopefully be able to play around with when it goes live in the public domain. I'll come back to that, let's say. I think I've got probably a couple of minutes, so we'll see how we go. And the recommendations out of the contract that we ran last year, obviously it goes without saying, we want to get more data. We're going to be hopefully working with more local record centres and local fungus groups. We're hoping to talk to more mycologists, field mycologists particularly there. We're going to use social media a bit more, and I've been working with people like Simon Harding to see if we can, as an ask, to go out on things like Facebook, the BMS Facebook page. We're still looking at whether the Kate database, we're going to look at that potentially, but we know there's a lot of duplication there. But we're also hoping that we can use the criteria to go to the Wildlife Trust to notify local wildlife sites as well. Okay. I'm going to race through this slide. There is more conservation action and recognition of fungi ongoing. We're looking at species in terms of re-evaluation as well. Although pink waxcap is common, really, in many aspects, it's still likely that the UK is still the centre of the population globally. So the argument is, should it actually still be a protected species? So we're revisiting that sort of thing. We're also looking at new species that are coming up, things like the Jubilee waxcap, which was only recently identified or given a proper name, if you like, and realising we may be the centre of the population, again, globally for that. So there's still pieces of work, and the database is feeding into that. And it goes without saying, the UK is internationally important for its waxcap grassland. We want to keep pushing that message, because at the moment, if you think about the number of sites I've potentially proposing for protection, we barely scratch the surface, okay? And just to quote Donald Rumsfeld, I think with fungi, this is probably as true a statement as there ever could be, quite frankly. It's true for fungi in general, it's certainly true for grassland fungi. And we really do need to keep going out there and finding out more information. That's kind of the end of my slide. I don't know, Emma, if there is a bit of time just to quickly show the map. Have I got a couple of minutes? I don't know.
Yes, we've got a number of questions in the chat. So please do share the map, because I think that will be really interesting for everyone. So let's do that first, and then we'll go to the questions afterwards.
I do apologise, that was a bit of a romp through, but there has been a huge amount of work and gone into this. So I've really tried to condense it tonight. So I do apologise for that rapid fire explanation of what we're doing. Okay, let's see if this will work. Hopefully it will. It takes a few seconds to open up. Anyway, you've kind of seen this map already, but what's happening here is it's now being populated with multiple databases, not just the FRDBI. If I zoom in, I will go to Adrian.
Sean, it's not. Sean, it's not currently working. Unfortunately, we can't. We can just see.
Is it not
a slide mapping of sites with chickadee assemblage? I think where you've clicked to open it, you need to share your other screen it's open
Got it.
on. Yeah,
is it open?
That's it. That's
open?
it. Yep.
Got it.
Thank you.
Sorry, apologies.
That's all right.
You have to be a whiz, don't you, at everything these days? So I'm going to whiz into or zoom into Hadrian's Wall, which I know a few of you know. And I'm just going to click on a dot. We can actually put on just the OS layer, but I'm going to just take it off for a bit of clarity at the moment. If we click on our map, it does take a... So Hadrian's Wall is a complex. So this dot is a centroid. It maps the Hadrian's Wall complex, and it shows the Chegg-D score there of 73, which is exceptional. What we can do is we can add the actual waxcap records. This is now a site-based look, and we can zoom in a tiny bit more. And this is now the one that Andy McClay, who I mentioned earlier, has mapped. And we can look at the data behind this to show the species that occur there. So he's got Hygrocybe fornicator there, or whatever its other name is. And we can start adding species to the list. So we're adding species all the time here to make the site information more powerful. We've developed a grid square. So this will be useful for partner organizations that will be able to use this map and then see, or hopefully see, where some of the hotspots are. And as we populate it, this will go a different color. And if I zoom out a bit, you can see the different colors which are showing hotspots on the map. And being quite quick with this, we can also, I'm going to take off that. We can then start assigning sites to priority habitat. So if you look here, you can click on a priority habitat. It says good quality semi-improved grassland. That's quite an important category actually, because good quality semi-improved grassland is not necessarily your lowland acid grassland or your lowland calcareous grassland. These might be grasslands that have been partly improved. These also happen to be grasslands where waxcaps and fungi might occur. So they're not floristically rich necessarily, this category. So we're adding the information all the time. We can put on the triple SI layer, which is then we can do further interpretation to see which sites occur within triple SI.
I think we've lost you, Sean. Can you still hear us?
So like I say, there's still, oh, have you?
Yeah, we've got you back now. Your screen's frozen slightly, but we can hear you.
Okay. Is the map gone?
The map's there. Your voice was gone temporarily, but it's back now.
Okay, that's good. That's good. Like I say, I'll leave it there because it just gives you a little bit of a flavour of what we're developing. And the final tool will be different to this. This is kind of prototype, but hopefully that just gives you a little bit of a flavour. There's a lot of powerful stuff behind here in terms of how the map's been put together. So, but like I say, hopefully that is of interest for people. Okay.
Fantastic. Thank you very much for sharing.
No, you're welcome.
So I've got a number of questions. I don't know if we're going to have time to go through them all, but let me ask a few of them. If we can't do them now, perhaps we'll come to them at the end. So let me just begin. When you were beginning your presentation, Jane asked, please can you comment on the role of Marasmius, or sorry if she says, sorry if it's been renamed. The common name is the fairy ring fungus. Yeah, thank you.
Sure. In terms of, I mean, we don't record it as part of the grassland fungi assemblage, just for a start. And in terms of how it interacts with other grassland fungi, I'll be honest, I don't know. Is the answer. There are lots of unknowns. That comes back to that Donald Rumsfeld quote. So I don't know the answer to that. But there are plenty of grassland fungi sites with the Chegdi that do not have the fairy ring champignon, for example. So that's as much as I can really say about that. Okay,
thank you. There were a few questions related to the records that were used, the data that was used to put together the database. So there was a question from Mark. Was there a record search on the local environmental record centres as well as FRDBI?
So the aim is to ultimately use all the local environment record centres. We used Sussex as a pilot, which Claire will talk about. We use Dorset as a reliable data source as well. And the ambition is to definitely go back to all the other local environment record centres to repeat that exercise. We have had some contact. I've had a contact with Cornwall, for example, to get further records. So that's a work in progress, basically.
Okay. And relatedly, do you get many fungi records from iRecord?
So yes, we do. And again, I leave that for Claire because the remit for Claire and Dorset and others was to do everything other than FRDBI, and her data largely came from the iRecord and other data sets. Yeah.
Okay, thank you. Liz asks, in the phenology info, is there any indication that Entomoma fruit earlier than other wax-capped grassland species and Geoglossacea later? Excellent question.
Only a slight hint. So I like Liz has actually said, I thought Entomomas occurred earlier, and I thought the earth tones were later. And actually there is definitely, I think earth tones are later. That seems to be from the phenology. But interestingly, I think statistically, and I haven't done the stats on it, but I think we will play around, there isn't real evidence to suggest that actually. In fact, I've just written a paper for Saim magazine. If we were directing people to do surveys, we would just say, if you can't do any other months, do October and November, because that is where the bulk of all the records appear to be. That obviously doesn't mean they don't turn up in July.
Okay. A question from Peter. Do you have a gut feeling as to whether there are any effects arising from fallow and red deer grazing on these sites?
Grazing is good. So I'm going to basically say that for the most part, grazing by deer or any livestock is going to be a benefit to grassland fungi.
From Gary, because of the intermittent nature of fungal fruiting, have you considered soil sampling and DNA analysis?
We're doing that as we speak, and we're just about to run a contract. In fact, we've just run a contract in the last year with Chris Knowles in Scotland, who has done the fruiting body survey alongside eDNA work, and it's something we're ramping up quite considerably at the moment as a tool actually for helping us monitor the triple SIs at the moment. That's how we're using it. Okay, yeah, definitely.
Nikita asks, are there any tips for looking for mushrooms in herbaceous communities, such as optimal conditions and soil type?
Oh, that's a good question. Are there tips for looking for mushrooms in herbaceous communities? Well, interestingly, we are trying to see if there is preferential vegetation types, and I will stick my neck out a little bit here and say that lowland or acid grass and types seem to hold the best assemblage, but quite frankly, those sites that have got acid grass and calcareous grass and neutral grass invariably are going to be the richest, and I think that's probably a reasonable assumption. Yeah.
There's a couple more questions, and I'm going to ask those now. Claire, you're just a little moment delayed, but I think that will be okay. So from Sam, when you were talking about the sites in the mapping, what kinds of places are these sites? Are they typically public parks, private farmland?
Again, a whole mixture, and that will be part of our further analysis. And as I said, there are always issues with access and how we actually ultimately produce this. Claire is coming up and she works at the Record Centre. I'm not a licensing expert in terms of how we get this data out here, but we've got people working on that sort of stuff. It's always an issue. But there are a lot of private sites here. So, for example, the Peak District National Park, we're able to gain access to a lot of private farmland and do a lot of good surveys there. So it is a whole mixed bag, but invariably there will be biases. What we've tried to do with this data is try and iron out some of that, if you like. And let's say there's people's gardens in here as well. So it's a whole mixed bag. But further work is needed to kind of see if we can pull out an actual trend there as well. Invariably, dare I say, National Trust sites tend to get very well recorded in comparison to everywhere else, it appears.
Yeah. Mark asks, did you only include recent observations of wax caps or also older observations?
Excellent question. In fact, the data, we had a cut-off. So this is 50 years worth of data, basically. So yeah, we use the FRDB DBI going back to 50 years, but we did do a little bit of analysis, more than just a little bit actually. There was a sort of shift to probably, I'm trying to be careful how I word this, the data that's 20 years old, we've kind of given it a slight preferential treatment, if you like, and there's going to be a little bit more work done on that, on the dates there. So we've gone back 50 years, but in terms of how we're assessing some of this data, we're kind of using a sort of 20-year in terms of our investigation of important sites. That's probably not a very good answer, I'm afraid, but we are definitely looking at how we're breaking down that date data.
Okay, thank you very much. There are a couple more questions. Hopefully we can get to those at the end. If not, would you be happy for people to send an email to you or to myself perhaps with their questions? Yeah,
and actually, Emma, what I would like to encourage people, we want to get data in, we want to get records in. Now, if people are happy to share that data with us, that's fantastic, accepting all caveats, you know. And we will put this out on social media as well soon, because the more data we have, the more accurate our assessments can be, and dare I say, hopefully we can protect sites in the future. You know, it's very serious. The point of threat is a very, very serious one for us. So, you know, that's the key driver behind this work.
Okay, thanks, John. If you've got specific website address or email that you want people to go to have a look for information, do you want to just type it into the chat for me?
I will do.
And then everyone can pick up from there.
Yeah. I've just got to work out how to stop sharing, actually. I've got it.
Okay, fantastic.
Right. I'm going to come off.
Thank you. Thanks very much.
No worries.
Okay, we're going to hand over to Claire now. So Claire, I'm just going to, you should be able to share your screen straight away. Can you do that?
Yeah, let's try this. Does that look like a presentation?
That certainly does. Thank you.
Great. Okay, well, thanks for having me this evening. It's a pleasure to talk to everyone. This is a cut down version of the talk that I gave at the BMS conference, so it should fit into the available time slot. So yeah, what am I talking about this evening? How I forgot about butterflies and learned to love fungi. That gives you a bit of a glimpse there of the sort of route I came into this, and it's not as a expert mycologist, so bear with me. I've been learning as I go along with the wax caps and grassland fungi. So I'm talking about local approaches this evening, and specifically in Sussex. And if you don't know where Sussex is, there it is. It's on the sunny south coast of the UK, and it's actually two counties, West Sussex, where I live, and East Sussex, and it's about 120 kilometres from end to end. So it's a fairly extensive patch, but small enough that one person can get to explore quite a lot of it over the years. So this is me. There's me nearer the beginning of my forays into the world of biological recording. I started off getting particularly involved in recording butterflies, and there I am on the South Downs admiring a wild continental swallowtail, which was a very memorable day. So just to give you an idea of where I'm coming from, I absolutely love Sussex, and my sort of entry into the world of mycology is as an amateur naturalist. I don't have any academic background in biology or mycology, so there's some aspects of mycology that I have found completely baffling, and really quite an uphill struggle to get into studying cells and microscopy and all of that, but I'm going to sort of talk a little bit about my journey and how I got into that. And I joined the BMS back in 2016. And before I got involved in recording fungi, I was particularly involved in this project, recording the butterflies of Sussex. We sort of coordinated an army of volunteers over a period of 5 years. I'm just going to move my thing around so I can see what I'm doing. Yeah, and it was a real lesson in getting to grips with biodiversity data. When I first took over as the volunteer recording officer for the Sussex branch of butterfly conservation, there weren't a huge amount of people involved in recording butterflies. We had this idea to do an atlas, but we weren't really sure if there were enough people interested to get out and survey over 1,000 tetrads in the county. But I think the idea of having a project gives people something to get involved with, and our experience doing that was, you know, once you start getting a few people involved and a few people excited, more people get involved. And over that 5-year period, we actually had more than 2,500 people contributing records to help us understand the distribution of butterflies in Sussex, and processed over 200,000 records, which went into producing the distribution maps for the atlas. So that was a kind of real lesson, I think, in the art of the possible, and also got me really excited about biological recording, and working with volunteer recording communities to improve the evidence base for Sussex. So when a job came up at Sussex Biodiversity Record Centre, I'd been working at the Environment Agency previously, but I thought, oh, this looks like an absolutely cracking job. I want to go and work there and manage a team that's providing ecological information services, making use of that data that the volunteer recording groups and lots of other people share with the Record Centre, so that we can make it available to inform decision making. And Sussex Biodiversity Record Centre is just one of many local environmental record centres around the UK. So a little perk of that job, we were based at the Sussex Wildlife Trust. So I hadn't been there very long, and I got invited to go out on a visit to Ebano Cricket Field. And this is what it looked like back in 2015 when I went there with Martin Allison there, who's a local mycologist, and the ecologist, Martin Allison, who's a local mycologist, and also Graham Lyons, who was the Wildlife Trust ecologist. And I really enjoy this photo now, because that look on my face is that I have absolutely no idea what's going on. I'm completely bemused by the fact that I've just seen a load of red and orange mushrooms that look all kind of the same to me, but there's Martin and Graham having a discussion there about which ones were crimson wax caps and splendid wax caps and scarlet wax caps, and it's just absolutely all mystery to me at that point, but it really piqued my interest, and I'm sure lots of people who've got into mycology will have had these moments themselves where they think, oh, this is fascinating and I want to learn more. So a bit later that year, I started my blog, and this was my attempt to really just get more involved in recording. I was really not sure at all what I was doing. So the blog was just a way for me to share what I was seeing and try and get some feedback from people on identification. And that was how my journey into mycology really began. Just having a go, one species at a time. And I joined the Sussex Fungus group, which were a fantastic and supportive group of people. There's Nick Applin there, who's the chair. He gave me a lot of feedback on what I was finding and the process of identifying fungi, and also run a field program each season, so you can get out in the field with people who know what they're looking at and talk you through the characters of fungi and the different features that you need to look for in order to arrive at an identification. And soon I was kind of seeing fungi all over the place. You know, you spend your whole life feeling like you hardly ever see any mushrooms, and then you start getting into mycology and you start spotting them everywhere. And these, which popped up outside my office, were my first wax caps that I found myself, a blackening wax cap there. And after joining the BMS, I went along on some of the events that they run. So I went up to Northern College in Barnsley, where Carol Hobart was running a microscopy workshop. And I remember finding some, what seemed to me at the time, fairly featureless orange mushrooms growing in the lawn there. But with the expertise of the people on hand, I was introduced to the characters of these species, like the honey scent that I could just about get off from that specimen. And went along to the BMS Fungus Study Week down in Cornwall, where we came across some more absolutely stunning wax caps growing in the dunes. I think this one was persistent wax cap, Hygrossby acuto-conica. So that's how I started getting into it and building up a feel for fungi. And it was that same year that this lady, Janet Whitman from the Natural England Field Unit, got in touch. She knew that I'd been getting interested in fungi, having a go, and she'd had this idea that the high weald in Sussex looked really promising for grassland fungi, and she came up with a proposal to run a 3-year survey, bringing in expertise from the Natural England Field Unit to survey sites throughout the high weald. But she wanted to run it as a partnership project, and give people the opportunity to get involved, and for volunteers to support the professional survey effort that Natural England was able to deploy in the area. And also what you can see Janet and her colleague doing there is collecting soil cores, because Natural England provided some funding for Aberystwyth University to do some soil DNA analysis, eDNA analysis, to compare the results of the fungal fruit body surveys with the results of those soil eDNA surveys. So I absolutely jumped at this chance. I was getting really hooked on grassland fungi by this point, and I said, yeah, I'd love to come out with Natural England Field Unit and look at some fungi. And as part of that, Andy McClay from the Field Unit ran a free training day, which was made available to volunteers from the fungus recording community. And it was Andy who introduced us to this quick learning key that had been put together some years earlier by a chap called Peter Russell, who was actually also Sussex based. And I didn't know him at all at the time because he'd moved away. He lives in the States now. But this is a really fantastic resource to start getting your head around, going back to that picture of me just being completely bemused about fruit bodies that all look kind of the same. This was a really handy resource to start getting your head around the different characters and what subtle features you need to be looking for. And when you pair this with David Bortman's book, then you've got a way in to really start tackling these stunning but often quite cryptic mushrooms. So that was my introduction to learning to recognize grassland fungi, and it was really just one mushroom at a time. It feels like it's still one mushroom at a time. They can be so variable. You really need to look at them and study them to get a feel for what you're looking at. And it was just a fantastic experience to get out in the field with experienced mycologists like Andy McClay from Natural England, because then, you know, when you find something, you can really appreciate what you're looking at. And there we're getting a great view of a fantastic big blue pink gill. Right, and around the same time, the Record Centre worked with the Field Studies Council to put on some training on using the FSC identikit. And people might have seen sort of multi-access keys that look a bit like this online. There's various popular ones for things like grasses and other, all different kinds of biodiversity. And I spent a day sitting in a classroom finding out how this works. And with the two things going on at the same time, it was a bit of a light bulb moment. And I thought, you know, if you put together that quick key with all the information in David Bortman's book in terms of the characters and the different parameters for these features, then you could put together an FSC identikit tool for grassland fungi, for wax caps. So I thought I'll have a go. It'll be a good learning project. So that was project number one. And this is online now. You can find it at the link there, or just type in grassland wax cap identification tool, and you should come across it. And it was a real it was a real deep dive into the variability of wax caps and their features, and also some quite tricky decisions for me as a relative amateur to tackle. So I had to make decisions about which taxa to include and which names to use. And actually, the fact that Natural England and JNCC had just published those SSI selection guidelines that Sean mentioned earlier made that really easy for me, because I thought, well, that's a fixed point. I can anchor to that list of taxa. I can anchor to those names. And I'll produce a tool based on that. And I was sort of producing it for myself as the audience at that time, as someone who wanted to get out in the field and identify what they were looking at. So I based it on field characters only, but there's a bit of a caveat there that, you know, with a number of different waxcap species, if you want to get to a confident identification, you do need to get the microscope out. So how that tool works, although it looks really fancy when you go on the website, the actual technology behind it has been deliberately made very simple so that you don't need a lot of technical expertise to develop a knowledge base and develop one of these FSC identikit tools. You just put all of that information, the parameters for the different species that you're interested in, into a big spreadsheet. So I did that. That was a big job. And I also documented everything I did, because I was conscious that there's a responsibility that comes with producing identification tools. If you make a bad identification tool, then you're potentially going to be responsible for a lot of bad identifications going out into the world. So every decision that I made, every kind of judgment call that I had to make, I documented that in this note. And I was particularly grateful to those people on the left there who were kind enough to read through this and comment on it and give me feedback on how I was putting it together. So I could be confident that it was kind of the decisions that I'd made were justified, and I wasn't going to unleash some horror onto the mycological community. And this is the result. So you've got all the different characters on the left there that I've included in the knowledge base. And if you click on one of those character headings, then you get a window like this with a bit of extra explanation of what those characters mean and how to interpret them. And if you click on one of the species, then you get information there. There's sort of information from that spreadsheet about the species and its different characters. And I also sourced images for each of the species so that you could get a visual aspect to the tool, a visual sense check of whether what you were looking at looks like the pictures. And this was one of the most time-consuming elements of the project, because sourcing critically identified pictures is really tricky. But I made a lot of use of this fantastic resource, which is the Danish fungus recording database, because you can not only can you find a lot of photos there, but you can also, with the help of Google Translate, read the commentary on those photos so that you can see which ones have been critically identified using microscopy, which species have been confirmed and accepted by David Bortman, who's the guy that wrote the book. And I thought, well, that's the closest we're going to get to very confident identifications and confident photos. So I sourced the photos from a whole range of different mycologists and people who are kind enough to all give me their permission to use them. So thank you to all of them. And I managed to get photos for every species of grassland fungi that's listed in those SSSI selection guidelines, except one. So that's my holding page for the one that I haven't managed to get. And that's the incredibly rare Hygrocebi aurantia. So if anyone's got a photo of that recorded in the UK, then please do get in touch. And then I did some testing. And I think any tool for identifying wax caps is going to have its limitations because they're so variable, they're so cryptic. And this was a collection that tripped me up when I was using the tool, because it was a particularly pale specimen of slimy wax cap, Hygrocebi irrigata. And in fact, this may be another species entirely, one that's a new species that's genetically distinct from the sort of typical slimy wax cap. But the feedback that I got from lots of people, and I think there's some people on the call here tonight who were involved in testing it, was that it's generally pretty good. It broadly works. It's just intended to be a support tool for identification. It's not always going to get you to a right answer. But I think some people have found it useful, and I certainly found it useful in getting me up the learning curve with grassland wax caps. So that's where I got to with that. And I had thought when I released the first version that I might update it following feedback from users. But my current thinking on that, and that's kind of informed by a sort of cautionary note that's in a footnote in the JNCC SSSI selection guidelines, is that they make the authors there make the point that names, we're in a period of relatively rapid taxonomic change. The organisms that we recognize within certain species concepts at the moment may be split up. So I think I'm going to wait and see how some of that taxonomic work on the grassland fungi percolates through, and maybe update this tool when all that's been done. But it feels like now's not the time to try and keep refining it. But it's there on the internet, and it's, I think, a good a good sort of representation of those concepts that are listed in the JNCC SSSI selection guidelines. So I'm always interested in people's feedback if you have had a go with it. So that was the first project. And I mentioned earlier that I really love data. So the more I started getting interested in grassland fungi, the more I wanted to read about what people had done before. So Sean mentioned this report that was produced by English Nature. I think this is the Shelley Evans report. There were a number of Sussex-based surveys that had been done and written up by Peter Russell and also Dave Bangs. So surveys of National Trust sites there, surveys of St Dunstan's Farm, which is a particularly rich grassland fungi site in the High Weald. A really comprehensive survey of lots of waxcap grasslands in Sussex by Peter Russell, and more surveys by Dave Bangs. Going back a bit, in 2004, the British Mycological Society held their autumn foray at Ashburnham Place, and they sent mycologists off to lots of grassland sites across Sussex to add to the evidence base then, and also went through records that have been provided by other mycologists in Sussex and sourced as many of those as we could and collated them at the Sussex Biodiversity Record Centre where I work. And that enabled me to work with the Record Centre with a bit of funding from the British Mycological Society to produce a waxcap atlas. So this is also online at the address at the top there. And you can use this tool to look at the distribution of waxcap fungi in Sussex. And I think the finest resolution that it goes down to is 1 kilometre. So you can see where the hotspots are, but also more importantly, the gaps. That's what I want to get people interested in. And it's quite exciting when you produce a tool like this to get people engaged with recording sites that have not been previously recorded. So I love it when I get emails like this about Wadhurst cricket pitch. So there we go. That's another dot on the map that we didn't know about before. And I think this atlas has been online for a couple of years now. And over that time, I've had people get in touch and share records for quite a number of different sites that were not previously known to the Record Centre. So, you know, it's been a motivation for people to add to that evidence base, which is really encouraging to see, and the point of it. And doing that work, I think perhaps brought Sussex to Sean's attention, and Sean got in touch with me in my professional role at the Record Centre to ask us if we'd be interested in doing a project with a fairly quick turnaround, mapping mycologically rich grassland sites in Sussex. So I said, yes, we would absolutely love to do that. And with some funding from Natural England, that enabled me to get one of my really clever techie colleagues at the Record Centre involved in mapping the sites, turning those squares that you saw on the previous map into sites. So another load of decisions about how to tackle this. Sean's already touched on the challenges and difficulties of defining a site, and we sort of took a starting point of sites that are already defined. So a lot of the grassland fungi sites in Sussex are burial grounds and churchyards. Those have already been mapped. Some of them are SSSI units, local wildlife sites, National Trust sites. They're already mapped. And then there's some other ones where we used Ordnance Survey master map to define those sites and put a boundary around them. And that's where the licensing issues come into play that Sean mentioned. We needed some thresholds to sort of work out how we would categorize these sites. And we just worked with records held by Sussex Biodiversity Record Centre at that time in order to sort of provide another perspective on working with fungal records data. So those are the thresholds that we used, and the thresholds for national importance are taken from those JNCC guidelines, and we sort of looked at comparable projects had been done. I think there was one done in Dorset, and used the thresholds for regional importance, the same thresholds for regional importance that were used there. And we had a bit of a discussion about thresholds for local importance, and there was more of an element of expert interpretation there, because 4 species on their own wouldn't necessarily show that a site was important, but if they were 4 particularly rare or high diversity indicator species, then that might give you an indication that that site is of local importance. And we worked with Martin Allison, that local mycologist who I showed you at Ebony Cricket Pitch, to do this work with us and help us interpret the records and the analysis that we were doing. So if you remember Ebony Cricket Field, that's what it looks like in summer. And that's what it looks like in the mapping of the waxcap records that we did. It's that little sort of oblong bit on the left-hand side. And when we came to do the analysis, we found there was 17 species of waxcaps that had been recorded from that site, which puts it firmly in the sort of regionally important category. But actually that site has no conservation designation on it at all. There's nothing in sort of planning terms that would let you know that that site is important for grassland fungi. So I'm running out of time now, so there's lots more I could say about that project and lots more examples, but by looking at the sites in detail, looking at them from the sort of local perspective and making use of all those additional local records that we had, produced not a good starting point for identifying which sites are important for grassland fungi in Sussex, but also lots of ideas for what else we should be doing, more work there is to do, working on those gaps, doing more surveys, getting more people involved in recording and monitoring. And Sean's already talked about what he's doing in relation to some of these other recommendations, about, you know, getting these sites recognised as priority habitats, exploring options for getting these sites recognised through local wildlife site systems. And I think it's also important to think about how we can share this information with site owners and managers to encourage positive management of these sites, which we haven't even really started to tackle yet. So we're pretty much at the end now. What next for me? Well, a few years of getting involved with grassland fungi has brought up lots of identification puzzles. Here's one from the other year, and I've worked with Sussex Fungus group and their DNA barcoding project to try and investigate this find, because it didn't quite look like meadow waxcap and it didn't quite look like pale waxcap. It looked like something in between, and I think it might be a cryptic species that is being found through soil eDNA surveys, but was previously just recorded as a North American species. Lots of work for me getting to grips with all the other letters in the Cheg-D fungal assemblage. So I'm hoping to have some fun with that over the next few years. And also letting people know what they've got. And I think that this is one area where people like us, all of us listening to these talks on this call, you know, Sean's got his work cut out there, working with all that data for England, and working on getting sites designated and recognised. But I think the impact that people can have locally on the ground, people who know the sites, people who know someone on the parish council, people who care about the mowing regime, you know, getting involved in recording, knowing where these important sites are, and advocating for their protection is an area where we can all help. And that's it. So a little bit of time left for questions, Emma. I will stop sharing.
Fantastic. Thank you very much, Claire. That's brilliant. I'm just waiting to see if anyone puts questions in the chat, but I following on from what you were just saying, I had a question for you actually. So as you were going through your talk, it's quite clear that you've worked, collaborated alongside and been supported by so many different people to bring together different expertise to help with the projects. And my question is really, how do you go about identifying the right people with those expertise? Is it through natural connections, through the way through your work? Do people step forward because they've heard about what you're doing? How do you go about finding the right people?
So a lot of it came from engaging with the BMS, going along to BMS events, and the BMS is quite a small society, but it's got some really eminent and knowledgeable people in it. So if they sort of see you working on something that has potential, then I've certainly encountered a lot of support and encouragement for what I've been doing. For me, social media was also a channel for connecting with people. I used Twitter quite a bit, and I wrote that blog, and some people, I'd get feedback from people through that. So I think John Dunkelman, who's on the who I saw on the call earlier, he gave me a lot of feedback, but I only really have interacted with him through social media, and also joining in with my local fungus recording groups, getting out in the field with them, got a lot of support there. And again, the more kind of contact you build up, and the more that people see that you've got a commitment to, really learn about something and try and do it properly, the more they're willing to help you. But it does take time. Yeah,
brilliant, thank you. So I'm just going back to a couple of questions that we had from earlier, which I think I'm going to bring Sean back in, actually. If I bring Sean, you back in as well. If you want to unmute, Sean, and if you both probably might be able to address some of these questions, actually. So I'm going to go back a little bit. Bear with me a moment while I do that. So Gary earlier said some years ago, I had a look at Rothamsted Park grass and found Punicia and others on the untreated bit. Has anyone looked there since? So that was Rothamsted Park. Do you know of any records there?
That's a good question, actually. I did know of Rothamsted, actually, so I think it's a place I've got to revisit. So, and I don't think, I haven't done a thorough search yet of the database. So I hold my hands up, as Claire has very kindly said, I have got a lot of work to do here. But Rothamsted will definitely be one to look at, because I was aware of the Punicia, definitely.
Okay, good, thank you. I've got lots of questions here, so just bear with me while I whiz down. Martin asks, anyone tried to create new fungi grasslands in the way we might make new lowland meadow communities, or adding rarer species to an already good fungi site for conservation purposes?
Shall I go, Claire, to start? Well, it's an interesting question, actually, because if only it was that straightforward. I've actually been working on a project to do with irreplaceable habitats, funnily enough, and I'm pushing very hard to make waxcat grasslands an irreplaceable habitat, because unusually St Dunstan's farm has had some interesting history and may have had more disturbance than most sites. But what we have found, and certainly that nearly all the grassland fungi sites, except for these few exceptions, are highly stable, undisturbed sites. And the best sites, interestingly, Andy McClay's work in North East England on some of the Victorian reservoir banks, which are 150 plus years old, are developing very, very interesting mycota. So our feeling is, and I think there's Dutch work done on this as well, that these grasslands take an incredibly long time to develop a rich mycota. And there's probably no better example, actually, than ancient cemeteries and churchyards, which, you know, they've obviously been there for centuries, a lot of them. That's not to say that grassland fungi will not colonize more recent sites. And there definitely is a series of pioneer type species. And like everything in nature, it's never that straightforward. But I think people have tried spreading spores around, mincing them up in food processors and throwing them out on sites. But as far as I know, with very, very limited success, unless anybody can disabuse me of that, these sites are important because they're showing us, telling us something about ancient grasslands as well, or certainly grasslands. And there will be interactions in terms of soil, pH, etc., and nutrient status, all those sort of things are interacting. Again, lots of research areas to be looked at potentially.
Thank you. Question from Ed. Do you rate and take records from iNaturalist?
Claire.
So iNaturalist is an online recording platform that's geared towards capturing observations, and that makes it distinct from iRecord, which is focused much more on capturing people's records. So there's an assumption with iRecord that when someone submits a record there, they've gone through that process of identifying it. iNaturalist works on a different assumption. Some people using iNaturalist will have gone through that process of identifying what they've seen using identification resources. Other people might have just uploaded a photo. So that makes iNaturalist data a much more mixed bag to deal with, and it makes it more challenging for verifiers and data users to assess that data and verify it and make decisions on whether it's usable for their purposes. That said, I think there's definitely a place for iNaturalist. I think there's a big place for people going out and making observations of grassland fungi that they see and taking photos and capturing them. And if you capture them with iNaturalist, then that information is geo-referenced. People can go back to that observation in future and see what was there. And some species of grassland fungi can be identified from a photograph alone, but many of them can't be identified to species. So I think it's got potential as a tool for capturing information on where grassland fungi are. But there are lots of challenges around turning that information into records that can then filter through to the kind of work that me and Sean are doing. So watch this space, because we've recently also, there's been a connection turned on between iNaturalist and iRecord. So some iNaturalist records are going through to the iRecord data warehouse for verification. So we're still in the early stages of that and seeing what kind of data is coming out of it.
That's really useful, thank you. One more question. I think we've got time for always lots of questions coming through, and we've just got a couple of minutes. I'll see if I can address some of them. As I say, if not, perhaps you'd like to email your questions direct to Sean or Claire. Claire, are you happy to share your email address in the chat for people to, if they don't get a chance? Okay, we'll just go to one more question here then. What could be the, this is from Sarah. Sorry, I beg your pardon, that's Sarah. What could be the impacts of fire on grassland fungi? Could there be an alteration in the diversity?
I don't know.
Honest answer.
I did lose you there, I'm afraid, for a second.
When I read the question?
Yeah, I can see the question on my screen, actually. Is it the fire one?
What could be the impacts of fire on grassland fungi? Could there be an alteration in diversity?
Honestly, I don't know the answer to that. And the one place that I know that does get regular fires, certainly on heathland communities, is the New Forest. And there's very rich mycota in the heathland and grassland in the New Forest. It would be an interesting subject to look at, but my feeling, my gut feeling would be that certainly in most of the UK, grassland fires, obviously we've just experienced the most incredible temperatures, etc., but are still relatively rare, and certainly autumnal and winter grassland fires are probably pretty much non-existent without somebody very deliberately, like in the New Forest, setting fire to the heaths in a controlled way. So I suspect we probably don't have any real life examples in this country to make an assessment on that.
Okay, thank you. If you're happy to hold on, there are a couple more questions. I don't want to make you stay beyond your time. So if you're happy to hold on, I will ask them. There are certain mushrooms. This is from Aniruddha. There are certain mushrooms which are host specific. So when you record the data, do you consider the grass species that are surrounding those mushrooms, or do you just record them as grassland species?
Speaking for myself personally, I don't currently record associations for grassland fungi, but the FRDBI does include the capability for mycologists to record associations with the fungi that they're recording. Probably depends on the mycologist. Yeah,
and I think, you know, we certainly ask for, if we are asking for surveys, I think I said earlier that we are identifying the priority habitat type, so whether it's a lowland acid grassland, and if we're very, certainly for our designation work, protection of sites, we will want a full plant survey done as well. So in fact, we could legitimately notify a site just for its grassland fungi, but we always do an accompanying plant survey as well. So it's not something that's necessary that we do per se, but there is fortunately some good data to show at least what sort of plant communities and plant assemblages are there. It probably needs a bit more work. I think it's a good question.
Okay, thank you. So both Sean and Claire have shared their email addresses in the chat. There's lots of comments saying thank you very much to you both for fantastic talks this evening, and thank you from us at the BMS as well. Thank you very much for both coming back and repeating your performance for us again online. It's been really good to see you, and thanks everyone for attending. Hope you've enjoyed this evening's talks, and hopefully see you again another time. Take care, everyone. Bye-bye.